Well, thanks very much for the possibility to speak here. It's a bit strange giving this recording a week in advance of the actual meeting, and it's a shame that we can't all be together and interact as normal. But anyway, it's an honor and an opportunity, I think, to present this work. So I'm going to give my special take on a multi-scale perspective of trying to understand the human cortex. That comes from, I think, a different place to most of the speakers in this session. And it comes through interactions mostly that I had with the group of Lars Muckli in the Human Brain Project, because we were in a project together collaborating. And that meant that the work that I was doing on rodents and what I think is closing in on a particular understanding of the way the cerebral cortex operates had to be married with fMRI, high resolution fMRI data. And that brought out, I think, a lot of interesting points about how to understand the cortex at different levels and how to integrate what essentially is our data on single cells with their data on more systems approach. And so in this talk, I'll go through what I think in the first part are the perspective changes that we came to through those discussions. And then I'll try to also elaborate on what the hypothesis is that we're bringing to our understanding of the cortex. And finally, I'll then present some new data on the human neurons that we've been able to do through tissue that we've got from excised tissue from patients. So, firstly, in terms of the perspective, there's a historical take on the way our view of the brain has developed, and because in the early days of neuroscience, there really was only the anatomical view, which focused on components and large-scale anatomy, more or less because there was no possibility to record from the components. And I think from the mid-20th century or so onwards, the focus changed to small-scale circuits and connectivity in the brain. And that's really persisted to today. So when one talks about models of the brain or a machine learning perspective, it's usually coming from a point of view that the connectivity is paramount. Typically the units or the neurons, the description of the neurons is relatively simple and is kept that way deliberately. And I think when we were trying to bring this perspective that my laboratory had with Lars's perspective, we came to realize that one needs to marry these two perspectives. Basically, we need to be able to put the components and the connectivity together to be able to describe exactly what's going on and to be able to interpret the various recordings that are going on. So that means that in principle for the cortex, we were very much focusing on the operation of the principal neurons, the pyramidal neurons. And I would argue we needed to get out of the dominant perspective of the integrate and fire neuron, both because this is really simplistic in terms of just the description of a point neuron, but also because it's not necessarily the case that the operation that's going on in a neuron is a simple summation function, which is nothing other than a kind of OR function. And in fact, there was another issue that came up that I wasn't expecting, and that is that the typical description of an integrated fire neuron, as you see in textbooks and in many places, actually is backwards to the way the actual components are arranged in the brain. So what we typically call input to a neuron is actually the output of the previous neuron. And so in some sense, the output of a neuron is really the release of transmitter. And this doesn't occur at the cell body. Typically, this occurs at the axon terminals and can be distributed far away from the cell body, all over the cortex in any layer that you like. And the same is actually true of input. This also doesn't occur at the cell body as is normally depicted, but occurs at specialized structures on the dendrites, which again, in terms of the cortex, given neurons that span all the cortical layers, could be occurring at any particular layer, no matter what the cell type that it's connecting to. So in order to understand fMRI data, it wasn't necessarily easy if you saw, let's say, activation in layer 1 to just conclude that this represents the activity of neurons in layer 1. This could be quite different. And in fact, if it really was the input of long-range axons hitting the tough dendrite to pyramidal neurons, then there's also now a lot of research going into expectations about how the neurons should respond to the input coming to the tufts of dendrites that has very special consequences that really aren't taken into account unless you know that in advance and interpret the fMRI with that in mind. So the other thing about incorporating the complexity of the actual brain is that it's by no means clear that neurons are best represented by single compartments or point neurons. In fact, they probably have multiple compartments, and it's not yet clear how many compartments, but it's almost certain that we should have more than one. And over and above that, it's not clear what kind of functionality we should give to all the different compartments, and in particular, what kind of activation functions or what they're computing should be assigned to these compartments. So we've got these multiple questions going on, which without being able to answer these, it's very hard to interpret anything any data that you get from recordings of the brain and the cortex in particular. We need to know how many compartments are in each neuron. We need to know the input-output function of particular neurons. And in that, that question actually focuses down on what kind of activation functions are intrinsic to the different parts of the neuron. And then once we've established that there are multiple compartments, we need to know possibly their new plasticity rules for multi-compartment neurons. And this is a long-range project, but all of which I would claim is really necessary. I can recommend at this point a recent paper that came out from the group of Jota Poiratsi in Forth in Crete, and they've got a very beautiful review article on ways of treating neurons and their computations. Anyway, I think over and above all of this, what one also needs is what I would call an operational theory, something which shows how all of these things interact, not just at the level of a neuron, but how how these more complex components operate within the network. And I had my own hypothesis, which I call the Dendrite hypothesis, which is now I think getting some maturity and to the point that we can start to make predictions and do experiments based on hypotheses about what we expect. Basically, the Dendrite hypothesis says that the pyramidal neuron is doing a very special operation in the cortex, and because it has two very separated compartments, namely the basal compartment and the distal compartment, that it's able to do a kind of coincidence detection on the types of inputs that come to these different compartments. We think that it's typically going to be feed-forward external data that would drive the proximal compartment, and a sort of internal stream of information that is perhaps a prediction or context that comes to the tuft compartment. And this is brought together by the intrinsic properties of the neuron in a very special way that we've labeled backpropagation activated calcium spike firing. But it is in essence then a mechanism for coincidence detection within the pyramidal neuron. And we think may be very crucial to understanding how the cortex operates. So we wanted to look at these in the context of the pyramidal neurons. Now, at this scales up, of course, and once you start looking at the cortex in this perspective, then there's a whole lot of predictions about what you should be seeing experimentally. In principle, we're predicting under different circumstances there should be dendritic activity, which translates to calcium in dendrites that in a modern context can actually be investigated much more easily than in the past. With tools like two-photon imaging and optogenetics, we actually have the wherewithal to test these predictions. And And I think in some sense we're actually closing a long loop, because I think the anatomists, they're all household names like Cajal, thought very much about these issues, but couldn't actually investigate them because it was so difficult to record from neurons per se, let alone do that in a behaving animal. So Cajal recognized that somehow the pyramidal neuron, in particular the human pyramidal neuron, was a very special entity. And this is from the Kuhnian lecture in 1894 that he gave in London. From the point of view of the differentiation and abundance of dendrites, enormous quantity of small collateral and terminal axonal branches, no other nerve cell appears to approach, even remotely, the cerebral pyramid of the mammals. And then he goes on to add somewhere else in the article, also in our opinion, it's very probable that the pyramidal cell possesses a very special intraprotoplasmic structure, which I take to mean that he guessed even way back then that there was what I think we would now call specialized intrinsic properties. And that's very much turned out to be the case, that these neurons are not just special in terms of their morphology, but they also have very special electrical properties. And of course, it should be said that they dominate the cortex. And that's another reason, I think, to suspect that understanding the cortical pyramidal neuron is very close to understanding the cortex itself. Well, in any case, I alluded to some of the successes that we think the theory has led to in terms of predicting experiments. And one of them was done by a very talented postdoc in my lab, Naoya Takahashi, who investigated what we call the moment of perception, which is that moment when you suddenly become aware of a stimulus, and you can manipulate this by changing the stimulus intensity such that you can go above and below the perceptual threshold. And here we had a specific prediction that when we had stimulus intensities that were below the perceptual threshold, you wouldn't see calcium spikes in the dendrites, but you would see them above the perceptual threshold and at the threshold. And this turned out to be true. And there's been a whole cascade of predictions that were able to be made on the basis of the theory, and that at the moment the data is totally consistent with this. I'm sorry to say that Noah is coming to the end of his time in our lab. It's been a great postdoc, and he's moving on to Bordeaux, and he's looking for postdocs, by the way, if anyone here is listening that is interested to go to a great lab with an exciting young PI. In any case, another thing that recently happened in terms of experimental predictions that worked out for us was looking at whole cortical brain states. And here we were looking at the coupling between the upper and lower compartments of the pyramidal neuron. And then using anesthesia going in and out of consciousness with anesthesia. And I won't go into the details here, but you can read this in the recent paper that came out. That essentially the upshot of this study was that we found that the anesthetics, through really specific molecular mechanisms, meaning really specific receptor types, decouples the neurons, meaning that the inputs that come to the distal compartment, the tuft compartment, don't get to the soma under anesthesia, and they can easily do this during the awake state. And what this effectively means from the perspective of the whole system is that cortical feedback is basically cut off by anesthetics and can't influence the system. So there's a whole part of the connectivity that goes away, and that this bridge, or this link, this nexus point is kept intact by ongoing thalamocortical feedback that feeds into particular receptors at this level. But the main point here is that what's going on is that we have a theory, if you like, that's making predictions that go from the cellular level up to the systems level. And I would be arguing that this is always necessary, and that you always need to take into account what's going on, the mechanisms and the complex mechanisms going at the cellular level, in order to have a hope of understanding what's going on at the systems level. Good. Well, in terms of the data with human neurons, which I'll turn to now, Here we had another fantastic postdoc in the lab, Albert Giddon, who focused on, well, we had an opportunity because we're working in a building at the Charité University Hospital in Berlin, and so we have access to resected tissue from human patients, mostly epilepsy patients, and Albert took over this project and was really the driving force for everything I'll show you now. And just before I show you his data, I should point out that the human cortex is about twice as thick as the rodent cortex, but in particular, that part of the human cortex that is disproportionately thicker than rodent cortex is actually the upper layers, and these are the sort of late developing layers. And here you find layer 2-3 pyramidal neurons that are more than twice the size of the rodent layer 2-3 pyramidal neurons. And so that all immediately raises important questions that we wanted to address. In particular, that we were then asking whether or not over evolution, whether there is compensatory mechanisms built into the human layer 2-3 neuron, because electrically speaking, this means that the distance from the synaptic input at the top of the cells, we nevertheless assume is really important, is much, much larger to the cell body. And so there was really two possible extreme outcomes, which would be that one would be that it's very much isolated and decoupled, or the other that there are interesting intrinsic properties to the pyramidal cell that compensate for this. In a way, it turned out to be both or neither, if you like. So we're basically asking, do we have the compact case of a neuron in the brain because of compensation, or do we have an isolated, decoupled neuron because there's no compensation? So the initial experiments, and I don't have a lot of time to go through all of the details here, so forgive me for going quickly through here, were done by doing dual patch clamp recordings in vitro. And here Albert did a lot of recordings from dendrites at all different distances from the cell body. And then he could test the propagation of signals in both directions. So either he could make a signal at the cell body and look at how that influenced the tuft, or the reverse, he could do a signal in the tuft and look at how that influenced the cell body. And the upshot is that the decrement in the influence of the other compartment is basically non-existent. So that a back-propagating action potential dies within about a third of the dendrite in humans, and it's even worse in the forwards direction towards the soma. So there's very little influence of depolarization of the apical dendrite at the cell body. And so essentially that meant that, at least for the passive responses of the neurons, there was no compensation. And that is a real conundrum, because in the end, we need to understand what the hell the input to the tufts are doing and how this can have any influence at all. But as the investigations went on, we looked at larger and larger injections into the dendrite, and it became clear that there were events going on in the dendrites that were very specialized, that we'd never seen before in rodents. So in the large scheme of things, we're not actually entirely sure that you can't see some of these effects in rodents in some circumstance, but in general terms, we have never seen such exotic and baroque behavior in rodent apical dendrites, in layer 2-3 neurons. And here's an example. If you look at this, its current injection is being injected into the dendrite, and that's causing the cell to fire. But before every action potential, in this particular recording, there's a little potential that's going on in the dendrite that we initially assumed must be a sodium spike because of its shape. And if you would ever see something like this in a rodent neuron, you can be quite confident that that would be a sodium spikelet or pre-potential. But it turned out that they were TTX insensitive, and then through further experiments we could see that in fact they were calcium spikes. So we ended up calling these dendritic calcium action potentials, or DCAPs. And in further investigation showed that they come in all shapes and sizes, and there's just a myriad of responses. Again, this will probably be an overload of information, but essentially these are all pairs of recordings from different dendrites and different neurons. And you can see on the left a dendritic response and the equivalent response at the cell body of that particular neuron. Here's another neuron, and so on. And without going into the details here, the main point I'm trying to get across is that there's a huge complexity in terms of the inner life of layer 2-3 human pyramidal cell dendrites, and so that made us want to investigate further the properties of these neurons. And so that was astounding enough, because I think essentially we've stumbled on a new type of spike that we could only, we've only noticed for the first time after looking in human neurons. But then there was another property that was potentially even more interesting, and that's that the decaps that can be generated actually have a sweet spot or a particular intensity where the amplitude is the greatest, where the response of the dendrite is the greatest. And as you go up in intensity, you actually get less and less response from the dendrite, which is unusual also in itself. So here you can see lots of examples from different neurons, and you can see that essentially they're tuned to, usually to the first the first potential that reaches threshold, and then they decrease after that. And if you average this over all of the different dendrites that we recorded from, then there's a very stereotypical activation function, if you like, of the dendritic compartment that we were looking at, and it's a very sharp onset and then a sharp decrease after that. And what this reminded us of was a function that could be used for a kind of an XOR operation. So I'll just explain briefly what I mean by that. There's two types of ORs. There's when in English, at least, when you use the word OR, you could mean either or both. So it doesn't matter if either or both of the outputs or inputs are on. You would consider that true. And then there's this so-called exclusive OR, which is just either or. And it turns out that many of life's problems are actually of the exclusive OR type. So when you come to the fork in the road, or you have a choice between chocolate or strawberry, you don't normally mean both. You very often mean one or the other. But it also is a well-known computational problem, because it's actually hard to build devices that compute the XOR function. So if you take the typical integrate and fire neuron, this is an inclusive OR operation, because basically one input might go above threshold and then it's on. The other input might go above threshold and you're on. And if both inputs are on, then you'll certainly go above threshold. So that would be an inclusive OR, whereas an exclusive OR requires that when both of the inputs are on, it would be off again. So we turned to a team in Crete, the group of Yoto Poirazzi at Forth, and another very talented postdoc, Nasi Papuzzi, and they modeled this in neuron. And essentially what you're seeing here is the outcome of quite a lot of work in terms of trying to realize this in a model of a neuron. Here we can have groups of synaptic inputs. So we're basically saying if there would be one type of class of event, this would make one type of or one distribution of synapses onto a particular neuron, and then another event would have another cluster of inputs, maybe overlapping inputs, and then there would also typically be inhibition, either ongoing or part of the stimulus, and so it was easy to show that you could have a situation where one input would cause spiking in the dendrite, which would lead to spiking in the soma, and the other input the same. But that if you combined the two, it would suddenly switch off because of the activation function in the dendrite. And counterintuitively, but in retrospect, actually, to be expected is that once you add inhibition, you can actually switch this on again. So you could regulate the extent to which the XOR function should work, or what kinds of combinations you would get. And so the XOR, I think, is just the tip of the iceberg in terms of trying to understand what could be computed by this. And in fact, in principle, there could be multiple compartments in this cell in a very flexible way, such that it doesn't have to be just 2 compartments. It could be, excuse me, turn my clock off. That's the end of my 25 minutes. So I'll just try and wrap up here. The upshot is that we don't know how many compartments it could have, but it's certainly more complex than an integrated fire neuron. And the activation functions, or the input-output of these, are very interesting and deserve further investigation. And I think we're right now at the beginning of trying to understand the plasticity rules that might be possible in this setting as well. And in the end, what we're dealing with is a neuron that, in terms of its length, looks very much like the rodent layer 5 pyramidal neuron, but has all sorts of interesting properties that will take some time to divulge and then integrate into some larger theory. But nevertheless, I firmly believe that it's absolutely imperative to undergo or undertake this project, if you like, of trying to understand the way these more complex multi-layer spanning units integrate at the whole systems level. So I really have to thank Albert Giddens for this project. He was really the driving force of the whole project. But also there was a team of people involved in the human experiments. There were anatomists, there were other people in the lab doing other kinds of experiments, helping Albert get on. And there were a team of clinicians involved that both procured the tissue from the patients and distributed that to other groups. And of course, the modeling team of Jutta Pairazzi and Nasi Papuzzi. And in fact, I should a shout out to the entire lab, who in various different ways were helping, not just with this project, but all the other underlying data in this. So that's all it is. Look, I'm not sure how the question time now proceeds, but thanks very much for your attention.